Hello everyone, my name is Eugene Baulin. I'm a postdoc at Janusz Bujnicki Lab at International Institute of Molecular and Cell Biology in Warsaw. And today I want to present to you our newly developed tool named CARTOIS, which means Computational RNA Topoisomerase. And what the tool does, I will explain in a couple of moments. So, as we all know, each year we keep finding out about new types of non-coding RNAs, which are functional and structured molecules and do not code for proteins at all. Usually, in the majority of cases, such RNA molecules form a specific structure that allows it to perform its functions, usually via binding with the different kinds of molecules, either with proteins, other RNAs, or ligands, or even metal ions. So the structure actually of such molecules defines their function. Now, let me remind you how we usually depict the structure of RNA. So usually we divide three levels of RNA structural organization, with first level primary structure being RNA sequence, which is a string and four-letter alphabet of nucleotides. Secondary structure is a set of base pairs, of Watson-Crick base pairs and Wobble base pairs usually, which can be depicted either as a planar graph or an arc diagram or a bracket diagram. And in case of arc diagram and bracket diagram, RNA sequence is stretched from left to right, and base pairs are shown either with an arc or with a pair of matched brackets. And the third level is a level of RNA 3D structure, which involves all additional non-canonical interactions formed on top of RNA secondary structure. And kind of surprising, actually, at least for us, that such a simple four-letter alphabet used in RNA can give us such complex and interesting structures, actually. So these are two examples of highly structured RNA, non-coding RNA molecules. The left one is twistor ribozyme, which performs self-cleavage, and the right one is flavivirus xRNA1, which gives exonuclease resistance to viral genome fragments. And both of these structures require the depiction of secondary structures here require at least three types of different brackets to depict the base pairs. So it's actually they are pseudonoted structures of third level, which is unusually high, but RNA is still capable of doing this. And it's kind of hard to believe that such complex structures could evolve simply by single point mutations. So we were thinking of any possible alternatives how the structures could evolve, and we found out we were thinking maybe there are some kind of RNA recombination events that could evolve a non-pseudonotous structure into a pseudonotous structure or something like this. Like here on two examples shown on the slide, you can see two possible paths of how this could happen. So on the left one, two different strands base paired with green nucleotides being glued together with a stretch of adenosines being cut out, and on the right, for example, like two strands base paired with each other form a hairpin together. And it turned out that actually we do know that RNA is capable of doing this, and we do know it since 1999, in the first paper on this topic from Chetverin Lab. And they described spontaneous rearrangements of RNA backbone topology as happening non-enzymatically, requiring only magnesium, and it was a rare event, so it happened only to one molecule out of 10 to the power 12 number of molecules. And we have more recent papers, like the paper from Lemon Lab from 2019, and actually the picture on the slide is from this paper. So they describe several various mechanisms of RNA performing such combinations, such rearrangements of backbone. And this year, actually, we have the first paper from Sasha Weinberg Lab, and it's the first paper where they discovered natural examples of non-circular permuted hammerhead ribozymes. Here you can see on the left the canonical structure of hammerhead ribozyme. It has three conserved fragments, block G, block C, block S, block G and block C are named according to the conserved structure sequence pattern. So the block G starts with G and block C starts with conserved cytidine, and block S is named after its inclusion of cleavage site. So having 3 blocks allows us to read the sequence in 6 possible ways. And so there are possible 6 different permutations of 3 blocks on a non-circular molecule. Yeah, so you can see all the 6 examples here. So it's like CGS, GSC, SCG, and 3 pseudonoted versions, SGC, CSG, and GSCS. So 3 of them are non-pseudonoted, and actually the canonical hammerhead ribozyme is non-pseudonoted, but 3 other are pseudonoted. And actually, why do we have 3? Because it's like a pair of triples, and each of the 3 are simply circular permutations of each other. Yeah. But between pseudonoted structure and between a pseudonoted structure and non-pseudonoted structure, there was a non-circular permutation. And in the paper they describe examples of pseudonoted cases of hammerhead ribozymes. So since we know the 3D structure of canonical hammerhead ribozyme, we were interested of finding such a permutation of backbone that could give us a pseudonoted structure out of a canonical hammerhead ribozyme. So we took a ribozyme structure from PDB entry 3ZD4, and we actually did identify a breakpoint at adenosine 28, between adenosine 28 of chain A and cytidine 14 of chain B. So if we cut the backbones near these 2 nucleotides and reconnect it in a different way, then we, from a canonical ribozyme of type CGS, we obtain a pseudonoted ribozyme of type CSG. Yep. So we can hypothesize that this is how pseudonoted hammerhead ribozyme evolved in the first place from a non-pseudonoted hammerhead ribozyme, or vice versa, we don't know. So actually the reason why only this year we have the first paper out there on non-circularly permuted sequences of non-coding RNAs is that it's really hard to study RNA permutations on a sequence level. And there are several reasons for it. So first of all, it must have taken place a really long time ago, at the times of RNA world, because now such events are not preserved at all, since if something happens to RNA, it cannot be then preserved at DNA level. So now we have DNA recombination and no RNA recombination. At least evolution is not driven by RNA recombination now. The second reason is factorial complexity of permutation search. So the possible number of permutations of a given sequence of fragments is it takes factorial time to search for. And the third main reason is really low signal. So in RNA the permuted fragments can be really short, and moreover we usually observe conservation for RNA on the structure level and not on the sequence level. It gives us a very low signal to search for. So we thought that maybe instead of doing a hard task, we can do an easy task. And an easy task would be to search for RNA permutations on a 3D structure level. So having 3D structure as input, RNA's 3D structure as input, we can find potential breakpoints and simply rearrange the backbone. So like this, in this hammerhead ribozymes, we found 2 strands of RNAs, non-neighboring ones, being close to each other in space. So we perform 2 cuts, reconnect the backbone, and have a completely new RNA sequence with possibly the same structure. And that's it. So we developed such a tool that can search for such permutations and perform them. Actually, we were thinking that so the tool finds all pairs of atoms O3' and P that are non-neighboring but close in space, permutes them, so reconnects them, but keeps checking that all RNA strands are still non-circular and still 5' to 3' directed. So we search for the simplest path of permutations that can give us a strand of the same length as the input RNA strand, but will be permuted. And this is a path of 3 consecutive permutations that give us a permuted strand of the same length. So we start with the sequence A, B, C, D, E, F, G, H, and we end up with a single strand reading A, F, G, D, E, B, C, H, with two intermediate steps. On two intermediate steps, we have a couple of RNA strands instead of a single one. So we developed a tool that can search for such sequence of events and perform the found permutations to the 3D structure being input, and we run the search on the entire protein data bank and cherry-picked the most interesting examples. So here you can see the first example being a SAM riboswitch. On the left, on the top part of the slide, you can see the original riboswitch, and on the right and on the bottom part, you can see the permuted riboswitch. The permuted fragments are highlighted in light color, and in zoomed in circles, you can see the site of permutations that took place. So you can spot that in the original version, you see a strange loop out in the yellow strand, which is absent completely in the permuted version. So if I would not know which one is the correct one, I would guess that actually the right one is the original one and not the left one. And you can actually also spot that the original pseudoknot is a permuted structure. It does has a pseudoknot. The original riboswitch forms a pseudoknotted structure, so it includes a pseudoknot, but in the permuted version the pseudoknot is gone. So we have a simple 3-way junction. So maybe actually the pseudoknot was obtained during evolution to obtain the additional structural stability to perform its function better, so to bind SAM ligand better than the previous version. Maybe we can hypothesize it. Then another interesting example is a couple of riboswitches, TPP riboswitch and THF riboswitch, the left one being TPP and the right one being THF. So you can see on the slide that the overall 3D shape of the molecules are close, but the coloring here, here I use a rainbow coloring scheme, so the 5' of the chain is in blue and the 3' of the chains are in red, and throughout the chain we have a rainbow order of colors. So you can see that the overall shape is close, but the topology colored here is completely different. Yeah, so this part is yellow, and this is not actually a hairpin, and in different colors. So the THF riboswitch is moreover a pseudoknotted structure, and TPP riboswitch is a 3-way junction. So they have completely two different topologies, but the overall shape is close. So we could find two permuted versions of them, a permuted TPP riboswitch and permuted THF riboswitch, with different permutations for each of the structures. And now you can see that not only the shapes of the molecules are the same, but also they have the same topology. Now the coloring is completely the same. This is the light blue hairpins, this is greeny yellow hairpins, and here we have the 3-way junction. So maybe these two riboswitches evolved from some common ancestor riboswitch via some of the recombinations like this. Maybe, maybe not. So actually, a really beam in the eye here is whether the permuted sequence actually would fold into the same structure as the original one. Yeah, so we hypothesize that after the permutations, we will still obtain the same structure, but on the permuted sequence, but it may not always be the case. So here is an example of permutations that we found in srpRNA. So the permuted fragments are in light color here again. So first you have the original sequencing structure, then a permuted sequencing structure, and then third on the slide here you can see an alternatively predicted structure, which is predicted by conventional computational tools pretty much consistently by different tools. So here I would bet that the permuted sequence won't fold into the same secondary structure as the original one. And now we have an ongoing wet lab experiments. So we performed some chemical problem experiments in the lab on the permuted sequences, trying to answer the question whether they would fold into the same structure as the original sequence, or they would fall into something completely different. So this is an open question still, and we are on it. So overall, the perspective of the potential use of our tool, Cartoiz, is like this. So Cartoiz allows us to explore the landscape of permuted variants. So having a 3D structure as input, we can find out all the possible permutations, tweaking different thresholds, of course. We can explore all possible permuted variants of our input structure. And then we have a theoretical interest and a potential practical interest here. So theoretically, we could hypothesize on possible evolution scenarios of how which permuted variant evolves from which. Or on practical side, we can measure something that we are interested in for a particular molecule, for like some functional efficiency or structural stability or anything other. And we can simply find out, identify the most stable or the most efficient variant out of our landscape that Cartoiz found. So this is the perspective and potential use of the tool, which we're really thrilled about. So that is it. Here I want to thank my PhD student David Bogdan and my supervisor Janusz Bunicki and all the other members of Bunicki Lab here in Warsaw. And especially wet lab members who are now doing all the hard work. The work was funded by EMBO and NCN grants. So thank you for attention. I would be happy to answer your questions if you don't listen to this as a record.